So I think let's go to the main question directly. They said discuss agglutination as a method of elimination of antigenic particles by the immune system of the body. All right. How does the body use what agglutination, or how does the immune system use agglutination as a method of what eliminating antigens? So let me say if this is a virus, okay, one method that the body will use to what eliminate this virus is what combining this virus and what antibodies. This is they binding together is what is known as agglutination. Alright. So it could be that it will now hold the antigens in one place for other immune stuffs to come and act on it, or it's now helping the elimination of the antibody, the antigen. Do you understand? So talk about it under the definition, mechanism of agglutination, types of agglutination, then roles in immune response. Then you get to appreciate the whole concept. For definition, we say that agglutination is a crucial mechanism, and this mechanism is it's crucial mechanism by which the immune system targets and eliminates what antigenic particles. What are antigenic particles? Things that are foreign to your body that have the tendency of causing you a disease. All right. So antigenic particles include things like bacteria, virus, and other pathogens. Okay. All right. So yeah, antigen bind with antibody. Okay. So the antibodies will help to hold them in one place. Okay. Then eliminate them. So what is the mechanism of agglutination? Agglutination involves the clumping together of antigenic particles through the action of what? Antibodies. So antibodies are combining with what? Antibodies are combining with antigens. Okay. All right. So antibodies binding to antigens. So these antibodies, also known as immunoglobulins, have a specific binding site that recognize and attach to antigens on the surface of pathogens. Okay. Then cross-linking. Each antibody has at least 2 antigen binding sites. Say, okay, 2 binding sites. Okay. So let's talk about it. Okay. So when multiple antibodies bind to antigens on different pathogens, they form cross-links, causing the pathogens to clump together. So it's basically resisting the motion of the antigen. You get the formation of large complexes. The result is what? There will be a formation of what? Antigen-antibody complexes. When they bind together, okay, they form antigen-antibody complexes, okay, which are more recognizable, and the immune system can now what? Target it. Okay. So what are the types of agglutination? Agglutination can occur through different immune pathways. It could be direct. This is when antibodies directly bound to antigens on the surface of pathogens, leading to clumping. It could be indirect. This involves the use of what? Particles such as latex beads or red blood cells coated with antigens or antibodies. So when mixed with a sample containing the corresponding antibodies, the agglutination occur. This one is just simply that process of trying to test for blood group. Do you remember? Okay, so that one is indirect. Okay, direct one is the one that occurs in your body directly. So what are the roles of this thing in immune system? All right, so they have several important functions. So it leads to what? The restriction of motion of the pathogens. That's immobilization. If you are moving, you are mobile. If you stop moving, you are immobile. Okay. So by clumping the pathogens together, agglutination prevents them from moving freely and infecting other cells in the body. Okay. Then enhances of phagocytosis. The large antigen antibody complex are more easily recognized. Okay. So phagocytic cells can now what? Act on them. Okay. Then prevention of infection. Agglutination can neutralize toxins and prevent viruses from attaching to and infecting host cells. It also facilitates complement activation. The immune complexes formed during agglutination can what? Activate the complement system, leading to destruction of these pathogens. You get? Alright, so clinical application is involved in blood typing. You know what I was telling you guys about. So agglutination reactions are used to determine blood groups by mixing blood samples and specific antigens. Infectious disease testing. Agglutination tests detect the presence of what? Antibodies and antigens related to specific infections. Serological assays. These assays use agglutination reaction to quantify the concentration of what? Antibodies in a sample or antigens in a sample. Alright. So that's A for what? Agglutination as a method of elimination of antigenic particles by the immune system.